Symposium. Hard to believe it's been 17 years. But we are excited to get started. We are also excited to have kind of a hybrid approach this year. So we have people in person, we have people online watching it live streamed on our YouTube channel. And so I think we have like a really good diverse audience, and even some people, you know, are watching that we're not seeing here in person today. Just a couple things real quick before we get started. We have a great variety of presentations this year, so it'll be today, tomorrow, and Friday. We have 27 oral presentations and 47 poster presentations. The poster session is this evening from 6:30 to 9. It's in the lobby of the Tom Ridge Environmental Center. Everybody's welcome to come back. We'll have some drinks, we'll have some hors d'oeuvres, you know, and there's going to be a lot of students here with their posters, so it would be great for anybody that can attend in person. That will not be live streamed, so that's a little more difficult to do, so please come back for that. If you are in person today, we ask that you do wear your mask. We have a mask policy in the building like other places do, whether we're vaccinated or not, so please try to keep your mask on. Just some housekeeping items real quick. Restrooms are right down the hall. Also across from the restrooms is room 108, so if you want to you know, have a discussion or meet with anybody or just take a break, you're welcome to use room 108 down there. After this session, we'll go into our lunch break. Lunches will be served in the cafe. They're boxed lunches, so you're welcome to stay in the cafe and socialize with other people that are here, or if you have to run back to class, that's fine as well. You can take the lunch with you. And then during the course of the symposium, we'll have drinks and snacks in the back, and just please help yourself. All right, I am excited to get started. Our first talks for the symposium this year, we'll start with Allison Burchell. She is from LECOM, from the Auditory Research Center, and she's going to talk to us about leveling up. Allison? If you can.
Okay, yeah, of course.
You guys will know if you need any water, and the microphone's right there.
Okay, so good morning, everyone. My name's Allison Burchell. I'm a third-year student at LECOM, and my talk today is called Leveling Up, a Glycinergic Allevogeniculate Projection that Circumvents the Inferior Colliculus. Okay, so to begin, I will take you through the ascending auditory pathway. So sound comes in through the ear as an air pressure wave through the ear canal and hits off of the tympanic membrane. And this will vibrate the middle ear ossicles within the middle ear, and this will act as a transducer to turn the air pressure wave of sound into a fluid pressure wave of sound. And within the inner ear, this fluid pressure wave will excite the inner hair cells in the organ of Corti, and this will send an action potential along the auditory nerve. The auditory nerve will project to the dorsal and ventral cochlear nucleus. From here, there'll be a bilateral projection to the superior olivary complex. From the SOC, there's a projection through the lateral lemniscus to the inferior colliculus. And this projection is bilateral, but mostly ipsilateral. From the inferior colliculus within the midbrain, there's a projection that's bilateral, but again, mostly ipsilateral to the medial geniculate of the thalamus. And from the thalamus, there's a projection through the auditory radiations to the primary auditory cortex. So this is the known and widely accepted ascending auditory pathway. And what this project focused on is a novel pathway. And this is one that's from the medial nucleus of the trapezoid body, located within the SOC, that goes directly to the medial geniculate of the thalamus. And this pathway was discovered completely on accident. And this pathway is defying this ascending auditory pathway, the known pathway, based on two things. So one, it's bypassing the inferior colliculus, which is known to be the obligatory auditory relay center. And two, it's a long-range glycinergic pathway directly to the thalamus. So what this project did was it did a series of experiments to provide evidence that this pathway does indeed exist. So I wanted to show you a closer look at what the SOC, or superior olivary complex, was. So this is a GEM-sustained section through the pons that shows us the SOC. So this is the collection of 6 nuclei here, which is located medial to the facial nerve. And what this project focuses on is the MNTB specifically. The MNTB is the medial nucleus of the trapezoid body. And this is a close-up view of the MNTB right here. And, And the MNTB is one of the 3 primary nuclei located within the SOC, and it's essential for sound source localization and temporal processing of complex sounds, such as vocalization. So we hypothesize that principal neurons of the MNTB have a homogeneous, long-range glycinergic projection to the ipsilateral VMG that bypasses the CNIC, that obligatory auditory relay center. So in this figure, this is the SOC, that collection of 6 nuclei located within the pons, and we're focusing mainly on this nuclei, the MNTB, and we're showing the projections to an MNTB principal neuron and the projections from that neuron. So it's receiving glutamatergic excitatory input from a contralateral cochlear nucleus globular bushy cell, and then it's sending glycinergic output locally to the ipsilateral LSO, MSO, SPON, as well as distant projections to the VNLL, INLL, and we know that it does not project to the DNLL, does not project to the CNIC, and then we hypothesize that it does project to the MG, so the thalamus, and that's what this project's focusing on. So this is what we did to prove, or to provide evidence that this projection does exist. So we used both male and female Sprague-Dawley rats aged between postnatal days 50 and 60. We did stereotaxic tract tracing injections, both retrograde and anterograde, with both retrograde and anterograde dye. So a retrograde tract tracer will be injected into a neuron's axon and will be taken up backwards towards the cell body of that neuron. An anterograde tract tracer dye will be injected into the neuron's cell body and will be taken forward along the neuron's axon. So for the retrograde tract tracing injections, we did both large and smaller focal injections of fluorogold into the diencephalon, which is the thalamus. And then we also did anterograde injections of BDA into the MNTB. 6 days after the injection, we perfused the animal and we sectioned the brainstem and counterstained it with neurotraced red. And select sections of brains, we did antibodies against calbindin, alpha-1 and alpha-2 subunits of glycine receptor, as well as vesicular inhibitory amino acid transporter. So we know that along the ascending auditory pathway, all of the projections should go to the CNIC. However, the MNTB does not. And we're showing this here. So an injection of the retrograde tract tracer fluorogold was made into the CNIC as depicted here. So it's being put into the axons of any neurons that are projecting to the CNIC and taken back up into the cell bodies of those neurons. And when we look at the section through the SOC, we can see that there is fluorogold labeling within the different nuclei of the SOC, so in the SPON, the BNTB. However, we don't see anything in the MNTB, maybe one neuron. So the MNTB is not projecting to the CNIC. However, when we do a similar retrograde tract tracing injection within the VMG of the thalamus, and then we do a section through the SOC, we see fluorogold labeling in over 50% of these MNTB neurons, showing that the MNTB is projecting to the thalamus. However, it's not projecting to the CNIC. So this is what's really cool and novel about this pathway, because it's defying what researchers and scientists thought was going on with the ascending auditory pathway. So this figure is showing us collectively our data from the large injections of fluorogold retrograde tract tracer that we did. It's showing us the injection sites and then the percentage of MNTB neurons that were labeled with the fluorogold. And then the same thing, but for the focal injections, so the smaller 60 nanoliter injections of the retrograde tract tracer. And notably what we found was that an injection that was mainly within the VMG provided us with the highest percentage of fluorogold labeled MNTB neurons, showing us that the MNTB selectively projects to the VMG. And then this is just another way to show what I just described on the previous slide. So when we look at the injection site and then the percentage of MNTB neurons that were labeled with fluorogold, we can see that an injection site that just included the VMG labeled the highest percentage of the MNTB neurons. And then the same thing, looking at the heat map with the injection site locations, an injection site including the VMG only provided us with the highest percentage of MNTB labeled neurons. So we showed that the MNTB projects to the VMG using retrograde injections, and now we're going to show that the MNTB projects to the VMG using anterior grade or forward injections. So this is a section through the SOC, a myelin section with the BDA. This is the anterior grade tract tracer. What this is the needle mark showing where the injection site was made, indicated by these little green arrowheads. And then this is the deposit of the BDA, the anterior grade tract tracer, into the MNTB. In this figure, this is just a depiction of the rostral pons, showing us the lateral lemniscus and the VNLL, so that we have an idea what we're looking at in this closer image here. And these little red lines are the BDA or anterior grade labeled axons. And it's no surprise that we're seeing labeled axons within the lateral lemniscus and VNLL, because we know that the MNTB projects to these areas. But what was really cool is that we saw these labeled axons within the thalamus. And when we quantified the diameter of these axons, we found that in general, they were a uniform size, indicating that they most likely came from a singular source. And finally, we compared the spatial relationship of the axons to the neurons that they came in contact with, and comparing it to a source that we know that the MNTB projects to, so the VNLL, and looking at how the axon comes in contact with the neuron cell body as well as the synaptic boutons. We looked at the relationship there and how it compared to the VMG and DMG. So in order to account for possible spillage when we were doing the injections, we injected the tract tracer into both the subarachnoid space and the cochlea, and we did not find any labeling of the SOC nuclei or the MNTB. So this was our negative control, just to make sure that when we do the injection, that the fluorogold labeled neurons were coming from the axons that were being labeled from the tracer being taken back up into the cell body, not from the tracer being spilled into the CSF and like these neurons picking it up that way. So the MNTB has different populations of neurons, and we wanted to figure out which population of neurons within the MNTB were the ones projecting to the thalamus. So we use Neurotrace Red as a stain to just show us all of the neurons within this section of the SOC. So all of these red stained cells are neurons. This magenta stain is fluorogold. So these are the neurons within the MNTB that picked up the tract tracer that was injected into the thalamus. And then these green neurons are the calbinin positive neurons. So calbinin is a calcium binding protein, and principal neurons of the MNTB are the ones that are calbindin positive, and this differentiates them from the elongate neurons of the MNTB, and the principal neurons are the ones that were glycinergic. So when we triple labeled these sections, we found that it was the calbindin positive principal neurons of the MNTB that were also fluorogold positive. So it were these neurons that were projecting to the VMG. So since it was the principal neurons that were projecting to the VMG, we would want to see calbindin positive axons within the VMG. And when we looked at the sections of the VMG that were stained with the calbindin, we did see calbindin positive on the low power view within the thalamus, and then on a high power view of the VMG that was counterstained with the neurotrace to see the VMG neurons. These little white arrowheads are pointing to calbindin positive axons that are traversing the VMG. So that was really cool. And then, so additionally, when we looked at the VIAAT, this is vesicular inhibitory amino acid transporter. It's located at axon terminals, and it participates in uptake of inhibitory neurotransmitters, including GABA and glycine. We found an abundance of this transporter within the VMG. And when we co-localize this with the neurotrace, so this pink are all of the neurons within the VMG, and we looked at the VIAAT within the VMG, we found that the VMG had significantly higher perisomatic and neuropil VIAAT labeling compared to both the DMG and MMG, so it has the capability to pick up the glycine and use it as a neurotransmitter. And then finally, since the principal neurons of the MNTB use glycine as its neurotransmitter, we would expect the VMG to have glycine receptors if the MNTB neurons are projecting there. And what we found is that the VMG does have glycine receptors. So when we did the glycine receptor immunohistochemistry, this is the yellow, we did actually see glycine receptors within the VMG. And here's a closer powered view. So the neurotrace, this purple, are the neurons, and then we see the glycine receptor here throughout the VMG. And then we just compared that to locations. So these are nuclei within the SOC that we know have glycine receptors, and that definitely looks similar. So we know that there are glycine receptors within the thalamus. So in conclusion, throughout these series of experiments, we have shown that the MNTB neurons, specifically the principal neurons of the MNTB, do project to the medial geniculate of the thalamus, specifically the VMG, and they do not project to the CNIC. The VMG neurons contain transporters capable of using glycine as a neurotransmitter, the VIAAT, and they express glycine receptors. And additionally, the VMG contain calbindin positive axons. So what can this novel pathway possibly be doing? So there are offset responses within the thalamus, and an offset response marks the end of a sound stimulus. So it's essential for sound duration encoding, and it has a profound importance for speech perception. And we hypothesize that the glycinergic principal neurons of the MNTB may play a role in generating these offset responses within the auditory thalamus. And then I just wanted to thank Dr. Plaza and Dr. Mansour. That's it.
Any questions for Allison? I do have a question. I'm going to try to make sure I word this correctly. So you were talking about like the glycine receptors is where the dye is picking up on. Is there any instance where an individual would have like lower concentrations of glycine receptors or higher? Would that influence your results depending on the individual and the amount of glycine receptors they have?
I guess that if there was a larger projection, you'd have more glycine receptors.
So then would that mean they would perceive the auditory information better or worse if they have more glycine receptors present?
So, I guess we don't really know what the pathway's used for, so I guess if it's used for the offset response, the glycine receptors would, I'm actually not necessarily sure.
Yeah, that's fine.
Yes.
First of all, thank you for explaining this well, because I have no idea what any of this is, and I feel like I kind of understood. Thank you. But what future research does this prompt you to want to look into? Like, what do you want to do next based on the results of this experiment?
So,
Did you kind of get all your answers?
I think what would be really interesting would be to look at the offset responses, like I mentioned on the last slide, but I guess in order to do so, you need additional equipment in the lab, and that would be something that we would need to do. But it's something that can definitely be looked at further to just investigate what this pathway is actually doing, because we showed that it exists, and it's something that nobody, yeah.
Yeah.
So.
Okay. Cool. Thank you.
We're going to end there because we're right on time. So thank you again, Allison. That was great.
Thank
And I
you.
agree with Sarah.
Thank you.
Our next presenter is Robert Waters, and this isn't his first time at Symposium, which is nice, so he's come back to talk to us again. And he is a graduate student at RegCom, and his talk today is Reinforcing Comprehension of Medical Microbiology.
Good morning, everyone. So this is going to be more educational research than benchtop research. So what we're looking at specifically is reinforcing comprehension of medical microimmunology with the use of online drawing workshops. So there's a need to enhance learning techniques to lead to a lot better retention and comprehension of material learned, especially in medical school. So specifically in the pre-clinical educational years, students had to learn so much information those first 2 years. They have to learn all the anatomy, all micro, immunology, biochemistry, etc. And they have to retain all that information for not only the first year, they go back to the second year, they have to get on the boards, they have to know that kind of stuff in rotation. So stuff they have to maintain and make sure they're going back and retain that information in order to understand what's going on next year and the following year and when they get to the boards. So there's a need to find different learning techniques and teaching techniques to enhance that learning and retention. So one such technique is generative learning. So generative learning essentially is where the student actually takes an active approach in creating their own type of study documents. So this can be student-written explanations, this can be students written like questions, peer teaching, things of that nature, where the students actually actively involved in their learning process. So one such thing also is drawing, and specifically drawing at the medical school level has not really been investigated that much. Specifically, there's been a few studies looking at anatomy, which you would guess because it's a very highly visual subject, as well as histology, but very little has actually looked at microbiology and immunology. So both of those subjects, especially immunology, is very visual as well. There's a lot of cell interactions, recognizing different cell appearances, stuff like that, that students have to know for their boards as well as during the first 2 years of preclinical education. So we wanted to introduce this to see if we do see effectiveness with drawing with immunology and microbiology. So this study took place last year, so everything was Zoom-based, as you guess. So we did this through online drawing workshops, and we had these students on Zoom and on breakout rooms. So we had breakout rooms about 6 to 7 students, and they work in groups to go over these different topics, immunology and microbiology, through the drawing workshops. And lastly, another thing we want to investigate is called the spatial contiguity effect. So that's mainly been looked at for authors' pre-made images, where basically it's saying that the closer the label is to a figure, the better the students are going to comprehend that information. So if you have a picture on one page and the next page you have the actual figure legend, it makes it much harder to understand that information because the student has to go from one page to the next to figure out what's this molecule mean, what's this molecule mean, what is it actually called. And that leads to a lot more cognitive load on that student. So it's harder for that student to actually understand what's going on as opposed to if you have the labels directly on the picture. So mainly all the research in spatial continuity effect has been looking at author pre-made images. None of the research has actually looked at student drawing and whether or not they label as drawings. Does that lead to that same kind of effect we see with pre-made or with the student-generated learning as opposed to author-generated learnings? So we wanted to investigate that approach as well. So the way we did this is last year in 2020, we had 191 MS1 LDP students at LECOM, and we had 3 online drawing workshops. Each workshop was an hour and a half, and there were 6 different drawing activities for each workshop. So our first workshop was only immunology, second one was only microbiology, and our third one was a mixture of microbiology and immunology. And we had 3 different assessments. We evaluated the effectiveness of these workshops, immunology quiz, exam 1, as well as exam 2. So there was a little bit of variables with the actual timing of these workshops compared to the quiz. We did introduce these through the curriculum, so we couldn't really change the days of the quiz and the exams. So they were there where they were. So for our first workshop, we had our quiz about 3 days after that workshop, and then they had the exam where they had the same material. They had to know the same material for that first exam about 10 days later. So they had 2 different assessments where we evaluated that first workshop. After we got through that, after each assessment for all 3 workshops, we did send them a questionnaire to get a gauge of how students felt the workshops were and whether or not they thought they were effective. And then eventually we got to the point where we collected the data, and we wanted to separate out the new students as well as the prior MMS students. So the prior MMS students actually are at LECOM for 1 year before they go into medical school, and they actually have a very similar microbiology and immunology course. So they're more of a master knowledge of the actual information in some cases. So we wanted to make sure that just because MS students are performing well, that didn't offset the data of the actual effectiveness of the workshop. So we looked at the students as a whole, but we also separated them out to make sure that it's not only effective for new students or it's not only effective for MMS students, but it's effective for both groups as well. After that, we analyzed the data and did a Wilcoxon test to determine significance. And here's for workshop 2. We just evaluated the effectiveness of workshop 2. 6 days after that on exam 1, same procedure. And then same thing with workshop 3, which is 5 days prior to exam 2. And so the way we evaluate the spatial contiguity effect is the best way we could think of doing this was actually randomly selecting half the activities the students had in each workshop to tell them, instruct them to label with actual words on their drawing, and the other one they're instructed to label with numbers. So that way, when you label with numbers, you actually have to look at that information, you have to look at the number, okay, so this is number 1, and you have to go back to that figure legend and figure out what number 1 was. Whereas opposed to, when you take into account the spatial contiguity effect, they actually label what number 1 actually is on the actual drawing itself. And then lastly, we did our analysis with a Wilcoxon test and deemed a p less than 0.05 to be significant. So here's an example of, with drawing, a lot of researchers have found that you need to give these students some guidance. If you just give them a blank piece of paper and say, hey, draw this mechanism of action of a toxin or something to that extent, the students get a lot of cognitive overload and they don't actually benefit from that drawing activity because they have to try to figure out what to draw, how to draw it. There's a lot of time just spent like how to actually draw this molecule. So a lot of research has shown that when you provide students with figure legends and those labels, it does lead to more effectiveness for those drawing workshops. So here's an example of one where we took account of spatial contiguity effect. So here the students were instructed to draw each of these parts as well as label those on the drawing itself. And so this is where we're looking at an endogenous infection or endogenous protein through MHC1 and antigen presentations. So here you can see this is a actual student-generated drawing. They went through the whole process of how we degrade endogenous protein and then eventually present that on MHC1 molecule to CD8 cells. So this was our first workshop during our immunology course. And then here is where we took account of looking at not taking account of the spatial contiguity effect. So here the student was instructed to label to each specific number on their drawing, and this is for specifically the Rabies replication cycle. And so this was part of our third drawing workshop, which had a mixture of micro and immuno. And here is what the student did here. So as you can see, they would draw like 1 as acetylcholine receptor, 4 as another molecule, 5, 6, 7, 8, etc. And so in order for the student to actually remember what each of those molecules are, they have to actually go back to that figure legend to figure out what 1 is, what 2 is, etc. And so here's where we don't take account of spatial contiguity effect. So this is where we compare the differences between actually labeling on the drawing as opposed to not labeling on the drawing. So now onto the actual data we looked at. So like I said before, we're going to look at a couple graphs for each assessment where we actually have all the students combined versus where we actually pull them apart, where we have prior immunology students and new students alone. So specifically, when we looked at the immunology quiz, we found that students performed significantly better on the questions pertaining to topics where they were reinforced during that drawing workshop as opposed to those topics that were not reinforced for the drawing workshop. When we then separated out between new students and prior MS students, as you can see on the board up here, once again, we found that the actual topics that were reinforced for these drawing workshops, they performed significantly better on those questions as opposed to the questions that were not reinforced through the drawing workshops in both groups, new and the prior MS students. We looked at course exam 1. Once again, we found that students that had the topics reinforced in the drawing workshops, they performed significantly better on those questions as opposed to the ones that were not reinforced. And we saw the exact same thing when we separated into new and prior students, where drawing questions, they performed significantly better on those as opposed to the non-drawing questions. So I forgot to mention this as well. So when we went through and actually did these comparisons, we actually looked at all non-drawing questions versus all drawing questions. We also went through and looked specifically, which is what this data is showing, is looking at, say, mechanisms of action for one toxin versus a question with mechanisms of action that did not include in the drawing workshop, had those comparisons. So that's what we're seeing up here, is actually comparing two different topics, but kind of the same thing. Like one's drawing, like one they have a question about EHEC, which they didn't draw, and the other one's like actual tetanus toxin, the mechanism of action, where they actually drew those out during the drawing workshop. And so we also, since exam 1 also had microbiology and immunology topics, we want to make sure that we separate those out to make sure that it's not only effective for micro or immuno, but that they're effective for both. So we separate them out, as you can see here. Even we only looked at the microbiology questions on exam 1, they performed significantly better on the questions that were drawing related as well as compared to the non-drawing questions. And then for the immunology questions, once again, we found that they performed significantly better on the questions that were involved in the drawing workshop as opposed to those that were not included in the drawing workshop. And then finally, with course exam 2, which is just immunology plus microbiology, because with our schedule the way it is, we actually come back to immuno again with immunodeficiencies, hypersensitivity, stuff like that. And we also come back to microbiology with viruses. So that's why we had to make sure both immunology and microbiology for course exam 2. But when we specifically looked at drawing versus non-drawing questions, once again, we still see a significant difference with the drawing workshop of number 3 of the drawing questions versus the non-drawing questions. And then here's the difference between new students versus prior MS students. And once again, we still see a significant difference between the drawing questions versus the non-drawing questions. And then spatial contiguity effects. So I just combined these all together just for time. When we looked at all 3 assessments combined, we also saw this for all 3 assessments individually, we found that the students performed significantly better on the questions where they were instructed to label with words when compared to the instructed to label with numbers. So indicating that spatial contiguity effect is holding up as well with student-generated drawings. The other thing we need to take into account as well is, obviously these topics are different, the questions are different, so we need to make sure that these questions have the same difficulty, they have the same validity to them. So what we want to do is look at drawing questions versus non-drawing questions, see if the discrimination index was different between those 2 groups. And what we found between the 3 assessments, immunology quiz, exam 1 and exam 2, that there was no significant difference between those discrimination index between those 3 assessments with those 2 different types of questions. And then lastly, we want to look at the order of questions. So we have first, second, and third order questions. We wanted to make sure there was no difference between the actual order of the questions the students were asked between the drawing and non-drawing questions. So you can see in red, these are the drawing questions, in blue you see the non-drawing questions. So with the immunology quiz, we found there's no significant difference between the number order, first order, and second order questions when comparing drawing to non-drawing. We found the same thing with course exam 1, no significant difference between first and second order questions between those 2 sets of questions. And lastly, with course exam 2, we once again found there's no significant difference between the first and second order questions between those 2 groups. So with that in mind, what we found was we knew drawing was effective. We just didn't know how effective it was at the medical school level, specifically with microbiology and immunology, and especially with it being online where we can't really control as much of what the students are doing, what they're looking at, as we can when they're actually in the classroom. So we found that even with the online drawing workshops, the students performed significantly better on all drawing questions when compared to the non-drawing questions, indicating that through these drawing workshops, they were group-based, they actually helped these students a lot retain this information. So we saw that, not long-term retention, unfortunately, but as far as short-term retention, the longest being 10 days. We also found that spatial contiguity effect does hold up with drawing, with student-generated drawing. So like I said before, most of the research into spatial contiguity effect is specifically looking at author-generated drawings and just comparing, like, if you give them a drawing with labels as opposed to, or an image with labels as opposed to one without labels, how effectively they answer post-test questions. Where we found that when students label themselves, it does lead to enhanced benefit. We also found there was no significant difference between the number of first and second order questions, as well as the discrimination index, indicating that as far as, we just want to make sure the questions themselves were not vastly different. As obviously, if we had much harder questions that were non-drawing questions, that would lead to a lot of confounding variables in that regard. And also, which I didn't include here, we also did surveys for each assessment for each workshop. And we found that the students did actually find this very beneficial. We had a lot of comments. We did a 5-point Likert scale on multiple different things, and across the board, the students actually liked the drawing workshops. We had about a N of 80 response rate, so obviously we had 191 students, but about 80 students actually responded to the questionnaires. And across the board, they liked it, which is very important because you want the students to be able to buy into this. Whether or not it helps, if they don't actually find it helpful themselves, they're not going to get much out of it. They're going to, do the least amount of work possible if they do not find this stuff helpful. But luckily, they actually found the benefit to the drawing workshops, which it also enhances including these into the curriculum of microbiology and immunology. And lastly, what I'm currently doing is actually looking at combining two different types of generative learning techniques. So specifically what I'm looking at is combining drawing as well as student-written explanations. So what I want to know is if we combine two different generative learning techniques, does that mean more benefit, or is that just going to lead to more cognitive, or more cognitive overload potentially? So I want to see if we can actually see a synergistic effect occurring when we combine these two different types of generative learning techniques as opposed to one or the other by themselves. So that's what I'm specifically investigating now to see if we can see that combination effect occurring, or is it just not beneficial at all. So that's what I'm looking at now. And with that, are there any questions? Yeah.
So you said that you did this in 2020, right?
Yes.
So are you going to provide these workshops again in 2021 to the students?
So we are doing them right now, actually. We actually just finished the first two workshops last week when we went through all microimmunology, like a week. So yeah, we actually went through, got it printed through Staples, so we have color pictures for everybody, stuff like that. So we went through that, and right now we're going through that right now. With they have a mixture of drawing, they have a mixture of student-written explanations, with various toxins and immunology topics and stuff like that.
Are they in person now, or
They are, yes, they're in person, yep.
Just figured I'd ask.
Yep, yep.
Thank you.
Yeah.
Do you think that there would be benefit to adding these components to the actual exams that the students are taking?
I mean, it could. That would be one way to look at some of these drawing effectiveness of drawing as well. It's just a lot harder to actually go through and grade, 200 students actually drawing on the exam. It takes a lot of time. Going through these drawings and actually looking through and make sure everybody turned them in, make sure, they had a rubric to make sure they drew them correctly. That took a lot of time, but actually do that for the exam too. And they want their, they want their grades back within like 2 days, and they start asking you questions every day. So I think it would be beneficial to actually see that as, to see if it does lead to more effectiveness, but it's really hard to try to do that on the exam sometimes. Yeah.
So did they start with an image, or did they just start with like a written scenario, and these are the pieces that you have to put into it?
Yeah, so they would get, they would be instructed to, like, for example, they got a figure legend, and they were basically told to draw out the process of endogenous antigen processing and presentation. So they were supposed to know that. They learn it in this class first, and then they come to the workshops to reinforce the material. So they should know it by then, at least an idea of it. And they know what to include then. They just got to figure out where it actually goes. So they're like, okay, we have a proteasome. Where does the proteasome actually fit in these steps of antigen processing for MHC1? And as far as TAP and how does it get into the ER and stuff like that. So they're given guidance, so they don't have to try to figure everything out just from their own. But they're given some guidance. And then we didn't really help them as much. We told them just work in groups, and if there's really big outstanding questions, we would help them with. But for the most part, we're just like, you guys can talk amongst yourself and try to figure it out.
So I think that's another factor too, is you said, the image itself, and then have them write about it, but then the discussion part is also.
Yeah, so this year too, we're doing group-based too, where it's, 5, 6 students working together, so they're talking amongst themselves, trying to figure it out together. So there's a little bit of peer teaching involved there too. Sometimes with 1 or 2 students that know more than the rest of the groups, they're kind of teaching the other students in the group. So I mean, there's some confident variables in this that we can't control for, unfortunately, but,
But that's all good.
yeah.
It's all
Yeah.
working well. Any other questions for Robert? Okay, thank you very much.
Thank you.
I'm just gonna stand here and read advance and just like have to read them anyhow.
Oh, that's fine.
Or at least look at what I'm doing.
Okay, I will let you move. All right, our next speaker, unfortunately, we have a little bit of a change that Madison Heater could not attend today. So possibly we might revisit that talk in the future, maybe if not this year, maybe next year. But the nice thing about being here at the Tom Ruth Environmental Center is that we have a lot of different partners that we work with, people that we work with. So I was able to run upstairs this morning and ask Holly if she could kind of fill this presentation gap that we have. Holly is part of the DCNR. She's a manager here, but she's also a graduate student at Gannon University. And so she is currently working on her research, and she's going to discuss what that research is today. So thank you again to Holly for, because I only told her this about an hour ago. So, but she's real good on the fly, and she's already done this presentation, so she's ready to go. So I'm going to turn it over to Holly, but thank you.
All right, so as Jeanette said, my name is Holly Best. I'm one of the assistant park managers here at Presque. I have been at this park since 2007. I've been within the state park system since about 1998. So I've been around a little bit. A lot of my work has been in resource management and natural resource management and kind of maintaining those natural populations. So when I had the opportunity to, go forward in my education and pursue a PhD, I chose to do that at Gannon in the Organizational Learning and Leadership program. And I was able to propose my dissertation topic here in May. I'm in my third year of classes of a 3-year coursework program. And I was able to find a topic that kind of bridged the gap between the social sciences and the resource management end of things. So I propose to do, and I'm working on, a multiple case study of natural resource practitioners performing invasive plant species control. Basically, I'm talking to some people who are killing invasive plants and seeing what kind of commonalities I can find. So, of course, the issue of invasive species is a very large one. It causes degradation of the natural areas. Invasive plants typically come in, they're very strong, they get a foothold in an area, and they just box out all the native vegetation. This also would have an impact on any of the native plants and animals and other species that would be utilizing those areas, because invasive plants do not provide what those native animals need. They also can choke out different areas, making it very difficult for native animals to move through areas such as wetlands, which is typically what I'm working in here at the park. They provide, as I mentioned, there's a threat to native, rare, and endangered species and overall biodiversity. A lot of the invasive plants that we have here on the park that I work with actually create just a monoculture of their species, so they just totally remove any sort of biodiversity. In addition to that, the economic impact is into the billions of dollars every year on treatment of these. And not only is the biodiversity attacked by these plants, but it has a big impact on the aesthetics and the view sheds. So if you think of a place like Presque, and we see 4 million visitors a year, and they're coming here to see the native habitat and to look and see what the shoreline looks like. If they are not seeing what should be here, that has a negative impact on our visitorship and also the economy and bringing people to this town. And that happens anywhere where people are going to see a natural environment. So in addition to that, the majority of treatments for these species are very expensive. So when you get to an area that is inundated, pretty much you have to use chemical to remove the species from the area. and that doesn't come cheaply. You have to have the chemical, the ways to apply it. Here at the park, we've even used helicopters, so that's not a cheap date to try to have a helicopter come in, but when you get to areas that are in largely wetland areas that are inaccessible by foot or land or boat, then air is the next opportunity for treatments. And the other thing is, touching on all of that, the big issue for practitioners is that there is not a lot of funding, there's not a lot of staff, and there is not a lot of training available. Mostly these issues are because, for instance, at this park, and any state park in Pennsylvania, there is not a line item for resource management funding. Pretty much the park has to carve it out of their overall operating budget, which means something else doesn't get done if resource management gets done, so that's an impact and that's difficult. We're fortunate here that we're within the Great Lakes Basin, so we can qualify and apply for Great Lakes Restoration Initiative funds, which we do, and that is what we use solely to perform the resource management at this park. If we didn't have access to those funds, we would not be doing what we're doing at the level we're doing it. Staffing is the other issue. There are no full-time staff in the whole state system of Pennsylvania. There are about 10 people who are identified as resource management managers. The rest of us all do this work as part of our other jobs. So it's just one little line item on my job description, but it takes pretty much my whole summer treatment season working on this program. So that means that other things are, getting pushed off a little bit or getting done in a different timeline. So something I identified within the literature, and it kind of ended up framing my study, is a concept identified as the knowing-doing gap. And this concept exists in a lot of different realms. So in the biological sciences it exists, in medical science it exists. It's basically a gap between research and practitioners in the field. There's something missing that's causing information to not go one way or the other way. And there's a lot of different ways that this kind of rears its head through the research. So it's a lack of communication, it's a lack of knowing what research exists, what pathways to that research exist, and it's also a lack of shared motivations. So some people are doing this work because they have funding, some people are not doing the work because they don't have funding. Politics can come into play, especially in areas that are, state or local or federally controlled. Different people have different goals and ideals, and that kind of happens where research may be focusing in on a certain part of this project and this process, and it's not giving the answers to the practitioners in the field that they need to do the work. And by the same token, the practitioners are doing the work, talking amongst themselves, but not feeding that information back to the research end of things to help build a stronger case for the work. So there's a lot of different things that are happening with that. So as I kind of touched on for invasive species and some of the background of the study, it's considered one of the main threats within the world for biodiversity, and that's well documented through the literature. In addition to the ecosystem impacts, which I spoke of, that also talks about how the aesthetics, which I mentioned, recreation, and even religious value. So some areas that are revered as their religious values are actually being degraded by invasive plants coming and filling in and taking over some of these areas. There's also another whole section on invasive animals, which I am not touching on any of this, but that is also a large impact area. And then the comment that it is estimated that in the United States alone, the annual impact of invasive species is in the hundreds of billions of dollars. And that was well cited in the literature. It's just the cost of doing business, and it is what it is running to get this work done. So going along with the rest of the issues, I touched on funding. That's a big issue. I did mention the Great Lakes Restoration Initiative. That's something that we have access to here in this park and amongst the Great Lakes states and into Canada. They also have access to those funds. It's a federal program. The thing is, if you can't prove a tie to the Great Lakes, you cannot apply for that funding. It comes in multiple different ways. It comes in, there's some pots that are given to each state to kind of dole out, and then there's competitive funds, which is what we apply for every year. We have been fortunate to receive funding for multiple years, and that is a large partnership amongst many partners that work out of this building even. So, there's also, as I mentioned, a lack of research in determining the needs that exist for resource managers to perform the invasive species management. There's very little research on this narrowly focused topic. So just kind of honing in on the knowing-doing gap, there's definitely a lapse in knowledge, and it's been identified as a knowing-doing gap. It's been talked about in the literature for about 20 years, and it's really kind of identifying the challenges presented to practitioners and just identified throughout the literature. And the other thing that was touched on a lot was there's just a lack of communication. You know, scientists speak a different language than field practitioners do. And I'm fortunate that I can kind of understand this and kind of understand this, so I can kind of bridge that gap a little bit. But most of the people I work in the field with would not even know where to look for a scientific paper on how to do something. They wouldn't even consider doing that. They would call their friend Joe and see how he has treated this kind of thing before. So it's really two schools of thought are being separated by this gap. So the purpose of the study is I'm doing a multiple case study, and I've already reached out and done some of my interviews. I've done 4 interviews, which is probably going to be what I max out at. I had proposed doing 3 sites within Great Lakes states. I ended up with 4. All of the resource management practitioners I spoke with are actively doing the work. They are from different backgrounds, hoping that, some of them have access to federal funding, some of them do not. Their programs are run a little bit differently. Some of them had designated staff, some do not. So I'm hoping to find some good comparisons as well as similarities amongst everyone I looked at. And just to kind of put it in a conceptual framework, in the center of this drawing, there's the knowing-doing gap. And that includes all those things that are listed there: research, modeling, field knowledge, best management practices, awareness, communication. All of those things exist within that gap. And then you have on one side the practitioners who are working on treatment methods. They have that field experience and that field knowledge. And they are also typically looking at a very broad ecosystem approach, because they know what they're doing in the field is going to impact everything else that is located within that ecosystem. Then on the other end, you have the research, which they're studying, which treatments are most effective. They're doing modeling. They're trying to determine, population studies and things like that. And a lot of the research is very honed into specific species, so it may not take into account necessarily the impact of that treatment on the entire rest of the ecosystem. So I kind of framed it with a central question, stating how do some natural resource management practitioners successfully navigate to be able to perform invasive species plant management. And then there were also a couple other sub-questions speaking to, how do they support their program, how do they acquire all their resources, being funding, staffing, training, education, and also how do they learn about best management practices. And those are the questions I utilized to write up the actual interview questions that I had when I did the interviews. There's just some definitions. If you're not aware, natural resource management is basically managing the natural resource, as it says. For the purpose of this study, I identified a natural resource practitioner as a person who's actively performing the control and management of invasive species. Invasive species is something that's not native to the area and it has to cause harm. It has to cause harm and damage to be classified as an invasive plant. And I also identified the Great Lakes Restoration Initiative, since I do talk about that in my interviews and in my proposal. So there are some assumptions for the study, is that everyone is doing what they're saying they're doing, and they're actually participating in this on-the-ground work currently. There's an assumption that the participants are going to be truthful in their responses to me and open with their communication, and that the participants hopefully will have an interest in providing valuable information that will inform the study and possibly, by an ends to a mean, close that gap. So there are definitely limitations of the study. With any multiple case study, it's limited by the number of participants. You're hoping that those results that you get are going to be applicable across a wide-ranging group of people. but you know that that may not happen, so the number of participants is limiting. There's also multiple variables. So, you know, knowing how this kind of works, there is not one program for resource management that works the same as another program. So it's going to be a little bit difficult knowing all the different variables and impacts that that program may be having. And also the findings not being applicable to all practitioners. So the significance of the study is in the lit review, there's definitely a void in the practitioner-focused research. It just does not really exist. There's a lot of scientific data, but it doesn't really go into the realm of, hey, let's go talk to the people in the field and see what they need and how they're doing it. So I think it's important to understand the aspects that enable the practitioners to perform in the field. Why are some people able to do this work and some people struggle through it or just choose not to do it? So what makes that different? And it's also important to understand all of the barriers. You know, you can read about them, but what are people actually saying is the problem from, you know, making them not be able to do this work. Additionally, it's going to hopefully inform the field of resource management to add to best management practices. I hope to be able to take this information and share that with the Bureau of State Parks, because there's only a handful of state park managers within the whole state of Pennsylvania who actively do this work. But hopefully, you know, some of this information will make it seem less challenging to people who are not currently doing it. And then also sharing information by doing presentations and reaching out to scientists and practitioners to try to close that gap. So finally, I just wanted to kind of give you an idea of how I kind of narrowed down the literature to show kind of the need for the study. So I started with just the basic concept of invasive species management, and that yielded over 41,000 results. I then added peer-reviewed, because that's of course what you would want to use. That brought it down to 23,000. So you can see there's a lot of like common knowledge pieces, not peer-reviewed items in this field. So I wanted to identify that. In addition to that, breaking it down to then invasive species management and social science, kind of bridging that gap, brought it down to just over 500 results. Focusing in on the term knowing, doing, gap, peer-reviewed within those results, brought it down to 511. But when I added the term environmental science, which kind of incorporated most of the field that would be doing resource management work, brought it down to only 38 results. So the majority of the knowing, doing, gap articles are not even within the realm of natural resource management. So kind of narrowed that down. And so the next steps for me are, I got my survey results. I have gone through and made sure that everything was written exactly how people said it. I've confirmed with them that I captured what they meant. So now I just have to sit down and start coding the data and figuring out what similarities exist, where there are gaps, where there is knowledge missing, and you know, then just write a little bit of a paper and get it turned in. So that's where I am. So I can take any questions if anyone has any. I don't know if I have many answers yet, but.
Any questions?
Jerry.
I didn't hear you use the word workshop at any point, but it seems to me that you can close that gap most effectively by getting the experts and the
Yes.
practitioners together.
Yes, and I have talked to my professor about that, and definitely that's something that, you know, because she's suggesting presenting at conferences, and I said too, I'm like, it would be really good if we can get involved in some workshops locally, or even, you know, statewide or within the Great Lakes.
Sponsored by the Regional Science Consortium.
There you go. Bridging the gap. So, yeah, definitely. And with state park managers, hopefully, you know, I have an audience there that I can tie some information with them too. I had actually thought at one time to do this study as a series of workshops and do a pre and post test, but that got kind of cumbersome with the process. So instead, I just kind of went back to my original thought to just do the multiple case study. Amber.
Do you think that once you have established, you know, how to best close this knowledge gap, that you could apply that to the knowledge gaps between, like, people teaching the public about aquatic invasives and people learning about it? Or I guess the knowledge gap would be more like people who already know about it versus people who are actively, you know, boating and angling, but they're not aware.
Yes.
They are aware, but they don't do anything. Do you think that that could apply to that situation as well?
I think it could definitely apply. I think, one of the big takeaways I think we'll see is how do we break down that information that is very scientific and make it realistic? And I mean, that's what we do as environmental educators, so we're like, oh, this is how you do it. But most people don't understand that, you can hear a scientific presentation and you will not understand 99% of it. So I think there will be some outcomes that will be able to go into other avenues like that as well. Anyone? Yes.
In the lines of invasive species education, I know that I'm from like southeastern part of Michigan, and I know there's a lot of things, especially when we talk about Belle Isle, a lot of different nature centers have kind of tried to at least break that kind of invasive species information down for kids and for students. How do you guys, especially if you're coming through getting funding and getting information from the Great Lakes, do you work with other groups along the Great Lakes when you're kind of looking at this knowledge gap and seeing how other places are kind of addressing it?
Yeah, so definitely being part of the funding source with the Great Lakes Restoration Initiative, a lot of us are members of listservs now and different, where people are putting newsletters in and things like that. So we're seeing a lot more of that information, but we see that because we're part of the network kind of, As far as education goes, we have used that funding to create some signage that is found out in the parking lot here at the Tom Rich Environmental Center. Some of our partners, the Regional Science Consortium, is a partner in that grant as well. So they created some signage that kind of explains different invasive species. We use the funding to fund our intern program. So shameless plug, but we're always looking for interns. You can apply with me till the end of January. Just send a cover letter and resume to my email. But they're paid positions. In addition, so we get the information out that way. Usually here at the park, we have a public meeting. I try to push the information out however I can. We have a Facebook page that is very highly visited. We have about 60,000 followers. So I put information on there about our invasive plants. Our education staff at this park has written multiple education programs that they give to not only the public, but also they train other environmental education specialists within the state of Pennsylvania and state parks to deliver this message and explain to people what we can do. So I think we're doing a lot to bridge that gap. I do wish that there was more connectivity amongst other Great Lakes partners, because I'm sure there's other models out there that are working really, really good that we just don't even know are happening. So.
I would definitely say that the Tom Rich Center does a fantastic job in trying to get this information to the public. I just think it's really interesting to hear, especially talking to other groups, maybe being able to pass maybe some of your information along to other groups and vice versa.
For sure, for sure.
Has any of this been presented at some of the continuing ed credits for licensed applicators to kind of get it out to that community as well? Or?
So I'm a certified applicator, so I go to a lot of continuing education classes. I have not seen many that actually focus in on the wetland systems. Most of the continuing education is how to apply herbicide, the basics of how to apply, how to safely do things, and then actually more like pest treatments, whether it's ornamental pests or insects. So the courses that I've taken within Pennsylvania, either through Penn State Extension or there's a couple other local groups, they really haven't focused in on the wetland systems. So yeah, there's definitely a way to get more information out there. I think they're knowledgeable about it to some extent, but it's not a training that is really offered, which would be beneficial because if you drive by any road in Pennsylvania, there is Phragmites and narrowleaf cattail along the whole way. So we need to do something, and some of it's growing into private yards, some of it's growing into other, public entities and things like that. So definitely knowledge would be power for treatment of that stuff.
Once you know it, you see it everywhere.
Yes, once you're calibrated, you can't miss it.
All right, well, that's all the time we have, but thank you, and an extra thank you to Holly for presenting on the fly.
Our next presentation right before our lunch break.
So on
Yeah, throw it in there. Right before our break, we're going to start our forensic session, so I figured that would be good. Start it right before lunch, and we'll continue it after lunch as well.
You still have an appetite
It's,
for lunch.
yeah, absolutely.
I'll try to fix that. Can we use this?
Yes. And that's forward, backward, and then the middle circle is the pointer.
Okay.
And so yeah, I'm excited to introduce Dennis Durkheim. He is from the Applied Forensic Sciences at Mercyhurst University, and he has been with us in previous years, so I'm looking forward to the update on this case. Dennis?
Hey, thanks, Janet. Good morning, everybody. I see a lot of familiar faces, students from our program. They've sort of taken over this afternoon. So what I want to talk to you about today is about forensic anthropology. I've been in the business for a long time, probably close to 40 years, and there have been a lot of changes. Forensic anthropology, essentially, as a discipline, started in the 70s, and one of my first forensic cases down in Pittsburgh was in the 80s. So I've seen a lot in the field, seen how it's evolved, and we're sort of at the forefront of how it's evolved, and it's changed pretty significantly to the fact that we claim now here at Mercyhurst that there has been a paradigm shift in how we view forensic anthropology. In the old days, it was basically you have some human bones, and forensic anthropologists are experts in human bones and extracting information out of that. And so we got involved in forensic cases sort of as a last resort. And so we were in our laboratory in our white lab coats, and we heard about a case, and then it would go through the system, and if the person was identified, said, okay, well, we'll wait for the next one. And so it was, in my eyes, very limited. My training at the University of Pittsburgh for my PhD included the physical anthropology, which is the study of bones and evolution and things like that. But it also included archaeology, and so I saw that there was an opening. There was a need for archaeology and the collection of data from outdoor scenes that the police were not trained to deal with. They were certainly very good at the indoor scene, but the outdoor, they had no idea. And so that's sort of been the focus of my career since then, bringing in forensic archaeology, processing the outdoor scene. And so that has allowed us to do a lot more than just sort of wait in our laboratory, wait for the bones to come in, maybe 1 or 2 cases a year. And so I can say that we here at little old Mercyhurst here do more forensic anthropology cases as an academic institution than anybody in the country. And we can include Canada, we can include so in the world, we do more. So we have a pretty good handle on it. So I'm going to talk about this particular case, and it sort of shows all the different aspects of what we believe forensic anthropology is today. So this occurred right down the road here in Frontier Park. Cascade Creek runs through it. Very, very beautiful little area. Let me go back here. So I'm sure all of you have been there. Very nice little gem here in the heart of Erie. So we are April 6th of 2019, and I think it was the Sons of Lake Erie are involved in a little spring cleanup. Get rid of all the garbage along the creek, so they had volunteers and a number of individuals, including kids, which is spectacular. Along the way, though, they found what they thought fat fingers. Along the way, they found what they thought was a Halloween prop. So it's sitting in the creek there, and so as the story goes, one of the kids, and maybe holding the stick there, had it on a stick, was coming out of the park, And Jerry Skripczak, who's involved with that, I don't know if he's a president or past president, he was there. He's a former Erie PD detective and all that. I worked on a number of cases with him. He said, well, wait a second, that is not plastic. That looks human. And so I was called down by the coroner's office. They were called down there, called down to take a look at it and said, yes, in fact, that is human. There's probably going to be more, or we have to look around. And in the meantime, the police, Erie PD, was looking there, and they found additional bones, which I was able to corroborate that were in fact human. And so then I called back to the department and said, we need to do a search of this area. And so we have a master's program in forensic and biological anthropology. So I had a number of students that could respond at the time. And so what we wanted to do was basically start at where that skull was found and search the area, see if there were additional elements. So here is just a line drawing of, and some aerial images of the park. Basically it flows south to north, eventually empties into the bay. There is a culvert, I have a pointer. There's a culvert here in which the creek is out in the open. It goes through a culvert under West 8th and then continues down, eventually almost approaching the extension to 79 there. So the skull was found in this, the cranium was found in this area. And so we wanted to search the creek there to see if there was any other remains. So at this point, in the old school forensic anthropology, we're down there. What has been found already is a cranium, tibia, ulna, and femur. So we have the head and an arm, and we have some leg. In the old school, we could say, well, we're forensic anthropologists. We deal with the bones. So we gather the bones, and we head back to the laboratory and try to figure it out. And we leave the cops there to figure out what's going on. But those are the old days. So what we do is, yes, we're experts in the bones, but we're also experts in what's going on in the outdoor arena. We're familiar with what happens to elements, to bodies out in the woods there, and so we're doing more than just analyzing the bones. We're trying to interpret, what happened in the past from those bones and some other aspects. So at this point, and we didn't leave it with just the police, because they'd say, what are we supposed to do? And so instead, we became involved in the recovery and the search for additional remains. And so the questions at this point is, who is this individual? That's obviously a very important one. And so we could have taken the remains back and characterized the cranium there and say it appears to be such and so, et cetera. But there are certainly other questions. How long have the remains been there? And that serves as a guide to trying to figure out who it is. At this point, we don't know. If the remains indicate that, it was a week ago that this person died and entered the creek there, then that will guide the police investigation of who it might be. Or if it's years later, then that will also determine who they're looking for. Where was the body originally? We have where the skull is, but is that where the body was located? We don't have. We just have three other elements there, and so we're going to try to figure out where that individual entered the creek, or if they did in fact enter the creek. They might have been on the side of the creek, and then some of the flooding episodes may have washed in. So we don't know a whole lot about that. And then certainly for the police perspective and the coroner's perspective is, how did they die? Is this just an accident? Could this be a suicide, or is this a homicide? Certainly for the police, if it's a homicide, then they're going to go all in and try to figure out who did this. So we're going to include in our aspect of the recovery. We're going to include the forensic anthropology, which is the bones. We're the experts in bones. We can extract a lot of information out of it. We're also experts in animal bones. So we're out in the woods, we're out in this, and so we have an expectation that we will find bones. White-tailed deer, dogs, things like that. So our expertise lies there, what we call forensic significance. And we do a lot of that. And so as a department, we don't do 1 or 2 cases. We actually do, on average, 110, 120 forensic anthropology cases throughout Pennsylvania, into Ohio, New York, etc. And so it's because we have these skills both in the bones, but also the forensic archaeology, which is the extraction of information from the scene. So we have methods to search carefully, systematically, to locate evidence. We locate it in its in situ position, and we document it, and we have the mapping, etc. And so we collect the information from the outdoor scenes very properly, and then the last part of that is something called forensic taphonomy, which is basically the science of what happens to a body after death. And so we are experts in that. We're about the only ones that are studying that, but that basically is what is the natural components to what is happening to the body. You know, it's not just what's happening to the bones, but it is, we're looking at the soft tissue. So we're experts in human decomposition and what are the factors that enhance it or delay it. We're familiar with animal predation on the remains. You know, what did they do? What marks did they leave? What do they leave? And how do they scatter it about? So what are the expected patterns of distribution there? Look at soils, impacts, look at the weather, environment, et cetera, et cetera, et cetera. So by the addition of forensic taphonomy to try to interpret basically the story, we have a cranium. We're trying to figure out the story between the time that we recover it to the time that that body, that individual dies or was deposited somewhere along that creek. So that interval is basically what we call forensic taphonomy. And so it's not just based on the bones. We can't get anything just out of the bones other than maybe some gnaw marks. We have to be familiar with, say, in this case, the creek, you know, how fast it flows, how often does it flood, what are the soils, what are the muds there, that's going to come into play, you know, how often do people use this park, all these other things factor into it. And so the main thing is, it's not just the bones anymore. Forensic anthropology, with the addition of forensic archaeology and taphonomy, is a different beast. And so at this point, we know that we have a human. From the skull, we can get some biological attributes from the bones. The biological profile, the skull, the head contains a lot of information. We can tell that it's an adult. We can tell that it's most likely a male. And if we have some time and take some measurements, we indicate that probably the ancestry is white. And so we have a good bit of information already related to that. We have some of the long bones. We can take those measurements of the long bones and come up with an approximate stature as well. So we have that already. So from those 4 bones, we have that. We look at them, is there any trauma? You know, you have somebody out in the woods there, oftentimes they're killed by some impact to the head, whether it's blunt force, a gunshot, something like that. In this particular case, there wasn't any trauma. That doesn't mean there's anything, the rest of it, but that's where we are at this point. And then something, the PMI, which stands for Post-Mortem Interval, is how long has the body been there? And so we have bones. And so we're familiar with approximately how long it takes for a body to decompose, to go from an intact body to just a bone, a separate bone. We have an idea from all of our cases that it takes at least a year, maybe 5 years would be, no, not 5 years, 5 minutes, right? About 2 years. So we have an estimate that this is about at least a year. Were the remains in the water alongside the shore and the burial? We don't know that. And so what we did is conducted a search. We looked in the water, and so we tried to do a straight line pedestrian search. We go upstream, we go downstream. We went upstream, downstream, along the side of the banks of it, and we found some remains. We mapped them in using a total station, and here, you can't see it, but it's a GPS unit. So we marked the exact location, because that'll factor in exactly what we're trying to figure out. And so in that first search, with us, we found 4 ribs, 2 vertebrae, fibula, mandible, metatarsal. So we found a good bit, and we were there about 4 hours. We also looked up through here. You can see here's where the culvert is, and then this part here isn't used by individuals, so it's tough going. But we needed to search, because one thing we tried to figure out is where the individual was deposited. The skull was here. The farthest upstream was in this location, was the mandible. So that would be likely the individual either started here or somewhere upstream, and so we needed to search this. We searched that, didn't find anything there. So like a history, and it didn't end there. So that was the first day. We found those bones, we recovered this, so that was a 6, so it was a good day. On the 8th, the police were still searching. They found some more material, 2 ribs, a scapula. On the 15th, somebody with a dog found a femur. How did we miss that? I still don't know how we missed that. And then the 18th, we decided after heavy rains to go back, and we found 2 ribs, we found more material. The 22nd, the police were looking again. They found a fibula. The 7th found 2 more ribs. And the 22nd, we went back, found a tiny bone of the hand. And then even August 20th, the tibia showed up. So it's a very strange case. We did a thorough search, but things still keep cropping up. And so you can see that we documented where we found material. The farthest was a hand bone, a finger bone way down here, but all along this path, and you can see the skull is originally here. So it's a very, very interesting taphonomic issue to try to figure out what's going on, but I guess just the flooding, et cetera, would move it out. We're trying to figure out where it started. The mandible is here. This is, like the remains aren't gonna go upstream from this location, and so this would be the likely start of it, but we didn't figure that out. So what we get from the bones, you can see here the assemblage. So we have head to toe, and so that indicates that the entire individual was there. Taking a look at this, calculated, take measurements and figured out aspects of this individual. One thing to note here is you can see the mandible was found separate from the skull, but it looks like it was sitting in the mud and staying there from the creek. And so it was sitting there for an extended period of time. And so this was our estimate of what we found. That it was a male, 30 to 55, white, a stature, etc. Didn't find any indication of trauma. 29 total bones, nothing was duplicated, it's just one individual. Suggests that middle-aged adult male, white male, and that stature. And nothing to indicate how they died. The other aspect of the taphonomy part of it is that the remains are disarticulated, they weren't cut apart, it wasn't dismemberment, it was natural decomposition. No soft tissue, which indicates a year. maybe even close to 2 years. And then it was deposited by the creek. And so we summarize that. There's a little bit of carnivore activity, but the estimate is 1 to 3 years is our estimate. The mandible and the femur were sent down to North Texas, NAMUS, for DNA analysis. We had dental, but there was nobody that they had an idea of who it might be, and so we had to do it this way. And actually, on November 4th, the individual was identified, a 44-year-old individual that had sort of been homeless, etc. And the thing, his family had not been in contact with him for years. So even though we identified him and we have all the other biological attributes, we don't know exactly when he died or the circumstances surrounding that. It's not liking me. Eventually, the remains were donated to our department for educational purposes, and so now his role is teacher to all of us. So, any questions about that? Again, I go on, and so this could be 2 hours later, but so I'll cut it short. Any questions? Okay, thank you.
Well, that concludes our morning session. If you are a presenter, RSC board member, or you served as a judge, you are welcome to help yourself to lunch. It is in the cafe, and so you can go ahead, and we will resume at 12:30 with our next session, continuing on the topic of forensics. Thank you very much.